Gottesfeld, who is going to speak about the HDACi inhibitors that are not yet at the clinical trial level, but promising treatment. Just a moment to change the configuration.
Sì, ho provato.
Okay, it works.
Thank you very much, Massimo, and I want to thank the organizers as well for the kind invitation to come and present here in Paris. Very much appreciated. First, a disclaimer. I am not a neurologist. I'm not even a neurobiologist. I'm a molecular biologist, biochemist, and dabble in synthetic chemistry a bit. So please, if I make any mistakes with regard to disease or neurobiology, I hope I have your understanding. My laboratory is located in that building right there, the Scripps Research Institute in La Jolla, California. And a few years ago, I read a paper from Bob Wells' group. Bob and his colleagues have studied the DNA structure that's adopted by a number of mutations in triplet repeat diseases. And specifically for the long GAA-TTC repeats that are found in Friedreich's ataxia, Bob has found that these DNA sequences adopt unusual DNA structures, such as triplexes, or a structure that he calls sticky DNA, which is an intramolecular association of these triplexes. And in the paper that I read that was in the Journal of Biological Chemistry, Bob suggested that one therapeutic approach to Friedreich's ataxia might be to find molecules that would either disrupt the unusual DNA structures or, in general, upregulate transcription of the frataxin gene. And it is transcription that's impaired in Friedreich's. And so this got me to think about potential therapeutic approaches. The first approach we took was the design of small synthetic molecules to bind GAA repeat DNA. And that, in fact, did work. We were able to design molecules that would bind GAA repeat DNA with high affinity. And these molecules would stimulate frataxin expression, mRNA expression, in cells derived from Friedreich's patients. But unfortunately, these molecules are rather large, and they only work in dividing cells. And so obviously for a disease, a neurological disease affecting terminally differentiated cells, this is not a good therapeutic approach. But around that time, a paper appeared from Richard Festenstein's laboratory, and Richard is in the audience. Richard found that long GAA repeats, when introduced into a marker transgene in a transgenic animal, would silence that marker gene, that marker reporter gene, in a manner similar to position effect variegated gene silencing that's found in flies. That's a type of gene silencing that causes heterochromatin formation on any particular gene. Heterochromatin, as distinct from euchromatin, is characterized for inactive genes, whereas euchromatin is active genes. In euchromatin, the histone proteins that the DNA is wrapped around are highly acetylated and have other types of postsynthetic modifications, whereas in heterochromatin, the histones are deacetylated and have other types of postsynthetic modifications. So an experiment that Liz Sarani, a postdoc at my lab, did was to look at the chromatin structure of the frataxin gene immediately adjacent to the GAA repeats by a technique called chromatin immunoprecipitation. where you use antibodies to the various postsynthetic modification states of the histones. And indeed, what Liz found was exactly what you would predict from the Festenstein paper, that is, that the histones immediately adjacent to the GAA repeats in the first intron of the frataxin gene, in a cell line derived from a Friedreich's patient, are hypoacetylated compared to the histones found in a cell line from a homozygous normal individual. Another hallmark of heterochromatin-mediated gene silencing is methylation of a particular lysine residue, lysine 9, in histone H3. And what Liz found was that in the Friedreich's cell line, she saw a high degree of histone methylation at this particular residue as compared to the active cell line. So taken together, these results suggest a model for heterochromatin-mediated gene silencing imparted by the long GAA repeats found in the frataxin gene in Friedreich's patients. That is, the frataxin gene in an inactive allele in patient cells has all the hallmarks of heterochromatin. We believe that somehow some proteins that initiate heterochromatin formation must recognize these GAA repeats, and Liz Saranya in my lab is taking a genetic approach right now to try to identify such proteins. Our model is that histone deacetylases that remove those acetate groups from the histones are found in the inactive gene as compared to the histone acetyltransferases that put on active chromatin marks on the normally active frataxin gene. So the balance between gene activity and gene repression between a normal allele and a Friedreich's allele of the frataxin gene might be modulated by small molecules that affect the activity of either the histone acetyltransferases as a positive effect or histone deacetylases. There are a number of chemical species known that inhibit histone deacetylase enzymes. And so what a postdoc in my laboratory did, David Herman, is looked at the different chemical classes of histone deacetylase inhibitors. And what he found was that among all the HDAC inhibitors that he tested, such as small carboxylates, cyclic peptides, hydroxamic acids, and benzimides, he found that one benzimide type HDAC inhibitor that's commercially available, called BML-210, would specifically increase expression of the frataxin gene in a